Hi everyone, I'm Caroline, a master's student at McGill University, and welcome back to Translating Cancer Research, a podcast series focused on discussing ways to more rapidly and effectively implement fundamental research findings into the clinic to improve the lives of patients with cancer. We host scientists, clinicians, industry professionals, and patient advocates to help us paint a picture of the translational research process and determine how we can optimize it. Before we get started, I would like to thank two of our sponsors. Our first sponsor is the McGill Center for Translational Research in Cancer, under which we have founded a student committee that is conceptualizing, hosting, and editing all parts of the podcast. The goal of this center is to provide a solid foundation based on state-of-the-art platforms, multidisciplinary training, and academic industrial partnerships for the advancement of scientific knowledge, specifically in this interface between fundamental and clinical research. Secondly, I would like to thank the Tremplin Consortium. Selected from a competitive pool, our podcast was granted the Tremplin Ambassador Award, which helped us acquire all of our recording equipment and software that we need for this show. The consortium reunites trainees across all McGill-affiliated research institutes with the goal of empowering biomedical trainees, strengthening community ties, and promoting a strong professional development infrastructure in the life sciences sector. This includes various funding opportunities, workshops, training programs, and networking events. For both the MCTRC and the Tremplin Consortium, if you would like to learn more about them, you can check the description below where we have linked their websites and LinkedIn pages. And now for the episode. So with us today, we have the pleasure of interviewing Dr. Marianne Koritzinsky, a leading researcher in cancer biology and translational medicine. Dr. Koritzinsky is an associate professor at the University of Toronto and director of research in the Department of Radiation Oncology. Her lab focuses on understanding how low oxygen levels in tumors affect cancer progression and treatment resistance. Through her work, she bridges fundamental discoveries with the development of new therapeutic strategies, highlighting the critical importance and challenges of moving innovations from the lab bench to the clinic. Thank you for joining us today.
Thanks for having me.
So to start off the episode, I would love to hear a little bit more about your backstory. So what inspired you to go into the field of research that you're currently in?
Yeah, it was a little bit of a convoluted journey, as I think it is for many people. I actually started out in my undergrad in doing physics engineering, and I really wanted to be a medical physicist. But then I developed a specific interest in radiotherapy of cancer. And I think it was, doing a little bit of master's work in radiobiology that I found that there just seemed to be just a vast space of undiscovered knowledge in the area of biology. And I'm sure there is in physics too, but it was more apparent to me when it came to the biology. And so that's really what made me spend more and more of my time actually researching the biological underpinnings of radiation biology and molecular biology signaling, and started my career in that area.
And would you say that there was like a specific experience that you had as a student that really, steered you into this direction?
I think there was a couple of things. It was also a bit of a special time in biology because there was a lot of tool development at the time. And so, for the first time, any lab could apply siRNA, to knock down a gene, which was new. And it really just opened up so much research that one could do in a much more kind of robust way. So I think that was one component. The other component was that for me, when I had started my PhD, which was a little bit more biophysics oriented, after about 2 or 3 years, I kind of decided that I needed some more inspiration and to expand my horizons a little bit. So I went to McGill and spent 6 months there visiting a biochemistry lab, which was very much outside of my comfort zone at the time. But I think, spending time in that other lab was really informative for me, not only about, learning about a new field of biology that I didn't know about, which was mRNA translation regulation, but also just to see how other people did science and how another group operated, right. I think was really influential and inspired me to sort of keep going in that direction.
Yeah, no, I think that's always something that's so valuable, is kind of stepping out of your comfort zone and going and seeing something else and seeing how other labs pursue either a similar area of research or something completely different from what you're used to. I think one thing that we are really interested in discussing today is the importance of fundamental research. So one element that makes fundamental research so difficult is making it clinically relevant to patient outcomes. So kind of given this, why do you think it's crucial to invest time and resources in studying fundamental biological mechanisms, even when the clinical application isn't immediately clear?
Yeah, I think there's a few reasons, we need to do that. First of all, I think we have to remember that all translational research is really built on fundamental research. We apply that kind of knowledge that we've gained all the time, even without really thinking about it. So I think that's a key piece. I mean, the other thing is that we don't know what we don't know, right? So I think there's certainly lots of examples where fundamental research reveals something that completely changes our thinking, and reveals new translational opportunities that we couldn't have imagined ahead of time because we didn't know what we didn't know. And so I think we have to remember, right, that if we only focused on translational research and trying to apply the things that we do know, then the pipeline would inevitably dry up. So I think it's really a key piece that we have to have in place in our research battery.
Right. No, I completely agree that without fundamental research, at the end of the day, you're not going to be able to have much of the translational aspect. And since we're on the topic of fundamental research, your lab does a lot of fundamental research and basic research. So do you mind sharing a little bit about what type of research is being conducted in your lab?
Yeah, sure. We actually do research along the whole pipeline, from very basic research to translational to also involvement in clinical research. But on the very fundamental side, we're asking a few super basic questions. And one of them is how cells, when they don't have oxygen, are able to secrete proteins. So I'll explain why we're asking that question. And it's because, so I'm in the cancer biology field, and cancer cells secrete a lot of proteins that are bad, essentially, for cancer progression. They mediate processes like migration and invasion, immune evasion, reprogramming of metabolism. But it's really unclear how that process works in all cancer cells, because many cancer cells have very little oxygen, so there's hypoxia in the tumors, which they tolerate very well. But everything we know about the biochemical process of secreting proteins tells us that it's reliant on oxygen. And that's because secreted proteins have disulfide bonds, so they have these covalent links between cysteine residues. And that is an oxidative process to introduce those bonds that as far as we know, requires oxygen. But it's very clear to us that hypoxic cancer cells secrete a lot of things with disulfide bonds. And so one of the very fundamental questions we're asking is, how are they able to do that? What serves as the oxidant, and what's the molecular machinery involved?
Very interesting, very interesting. Yeah, I know you had mentioned that some of them are more fundamental, and what exactly on the more translational side is your lab doing?
So on the more translational side, we have in the past we've studied or tried to really implement an idea that inhibiting oxygen consumption would reduce tumor hypoxia. And the reason we want to reduce tumor hypoxia is, as I mentioned, both because in hypoxia cells secrete a lot of proteins bad for cancer progression, But we also know that hypoxic cells are really resistant to therapies, and especially radiation therapy. You really need that oxygen around to cause the toxicity of the radiation. And so we had this idea that we could use actually a well-known anti-diabetic medication, which is called metformin, to do this, because it had just been figured out actually that what metformin does in diabetic patients is to reduce oxygen consumption in cells. And so that was interesting because metformin had been used for decades in diabetes, but its molecular mechanism hadn't been known until then, quite recently. And so we thought that, well, maybe if we gave patients metformin, then the cancer cells would reduce their oxygen consumption, and then that would increase the oxygen availability in the tumors and make them more responsive to radiotherapy. So we did the preclinical work, and that idea seemed to pan out in the preclinical work, and then it moved into clinical trials. So we published one very small clinical trial in cervix cancer a couple of years ago that was also positive. So it looked like women with that kind of cancer that weren't diabetic, but when were then given metformin, had better oxygenation of their tumors and fairly good outcomes at the end of the day. And that trial has now been replicated in other places of the world, and we're really anxiously awaiting the results from those trials as well to see if they're also positive.
Yeah, no, that's very interesting. And I think this is a really good segue to kind of discuss the challenges in translating laboratory discoveries to the clinic. And I think something that is on a lot of our minds, especially, fundamental researchers, is, okay, if you realize that you found a target that has translational potential, what exactly are the next steps moving forward, and what have you found in your experience were, the next steps after having, coming up with a discovery like this?
Yeah, I think that there are many challenges. I think we were very lucky in the work I just talked about, in the sense that there already was a drug that was well known and very commonly used that we could try to repurpose, and that is kind of the easiest scenario. If you want to design a drug, of course, we know that that's a very, very long and difficult road and very costly as well. But that is, I think, that is what many of us are doing. We have a couple of targets that we're working on that we have found to be specifically essential in cancer cells and not in normal cells. So we're trying to do some drug development against those targets. But it is very challenging space. It takes a very long time, and so you have to be patient and also try to fund that kind of research. It can be difficult as well. But I think one really key piece of translation, at least that in my experience, is also, to have a very close relationship with clinicians along the way, and not just when you've published your paper. That's too late to then pick up the phone and try to cold call a clinician and ask if they want to implement your research. So I've had very good experience with working in teams and in programs that have basic scientists and clinicians, so that also, we can have that clinical perspective when we do some of our research, how we ask our questions, what models we use, what would actually convince them to take that a step further later on. And I think that's been very productive, and is something that's extremely helpful as we develop translational research, is to have that sort of bedside to bench and bench to bedside mentality, and include each other in the research.
Right. I mean, at the end of the day, collaborations between researchers and clinicians is so important for translational research. I just want to go back to one point that you made earlier regarding these targets. I know your lab recently discovered the dependency for pancreatic cancer pancreatic cancer cells, I should say, on PRDX4, I believe it is. And kind of, if you wouldn't mind talking a little bit more about that, and because I know that was something that obviously is, has is the discovery is obviously very fundamental, and where you think kind of that might end up going.
Yeah, that was a few years ago now. So we were actually interested in sort of regulation of redox homeostasis in general in cancer, because it's been known that cancer cells produce a lot of reactive oxygen species. That's really a byproduct of high proliferation and high metabolism. And so they're very dependent on antioxidant systems. And some of the major antioxidant systems have been tried to be targeted before, but often associated with a pretty narrow therapeutic window because normal cells are also dependent on them. So we looked into functional genomics data for various antioxidant proteins, and we found that about half of pancreatic cancer cell lines in a panel of 36 was dependent on this one protein, PRDX4, which metabolizes hydrogen peroxide in the endoplasmic reticulum. So it's a very sort of specific function of it. And so we thought that, pancreatic cancer is really aggressive, and so if half are sensitive to it, that's pretty good. And we were encouraged because somebody else had already made the PRDX4 knockout mouse, which was healthy. And so that's why we thought that there might be therapeutic ratio associated with that. So in the paper, we elaborated on this story and described why some cancer cells and not others were dependent on it, and that had to do with the way that they reprogram their metabolism. And after publishing that, we have actually since then been working on trying to find an inhibitor against PRDX4. So we spent a few years, and this is just to illustrate how difficult it can be, basically a few years, probably 4 or 5 years even, trying to establish the enzymatic assay to do a high throughput screen to find inhibitors against the catalytic activity of PRDX4, and basically we're not successful. And so now we've turned our attention to other modes of drug development. So we are interested in trying, instead of inhibiting its catalytic activity, trying to degrade it. So there's new ways that have been developed in the recent years where, the idea is to find a drug that binds PRDX4 and then link that to another compound that binds an E3 ubiquitin ligase. And so then in that way one could target the protein to be degraded. And so that's what we're pursuing right now, and we shall see how it goes.
Where that goes, where that leads. And kind of in your experience, especially when it comes to, something like this, what exactly do you think, what do you find is the biggest barrier when it comes to, going from preclinical research to success in human trials? Is it, really finding a compound that's able to target a protein of interest, or do you think there are even, more extreme barriers down the line?
Yeah, I think, one fundamental difference is that we have to study this in model systems, and so there's limitations of our model systems, right? And sometimes there's not the success rate one would like in clinical research that's built on preclinical research, and sometimes perhaps there's just fundamental differences between an animal and a human, and you just can't get around that. But I think what we need to think about is of course the models we use, and trying to use several models, because in a way, when we do laboratory research, our power is that we can control so many parameters, and we can do robust research and make good scientific interpretations because we have controlled a lot of the parameters, right? But when we move into clinical research, there's a lot more diversity, a lot more variability that we have no control over, and I think that's also part of the issue. So I think one aspect is probably to have, try to get at it with more models. They will all have their individual limitations, but then at least we know that it is more generally applicable. But
of course,
but, very often our scientific system doesn't encourage that, because we like to have positive results. We don't like to try to prove ourselves wrong,
Yeah,
or
show that
to
maybe
find the limitations, right? Of
maybe
our work.
maybe it works in one model but not the other.
Right. And so I think we also have a scientific system that doesn't necessarily encourage, you know, trying to find the limitations, right?
Yeah,
So that's another, I think, fundamental reason why sometimes it doesn't always
right.
give the same results in clinic as well.
Right, another like kind of difficulty or hurdle that I think has to be overcome in the field.
Hmm.
Yeah, so something that you talked about were how important collaborations are between researchers and clinicians and even industry partners. So, how do you find mentors come and play into the importance for one's own self promotion in the field of let it be basic fundamental research even to further down translational research?
I think mentors are critically important, both for the way that we do science, so learning how to be good scientists, and also, as you mentioned, for more career development and learning some of those other skills that we need to be successful in science. I think that there's lots of amazing mentors, but nobody can be everything to everyone, and so I would really encourage young researchers also to look for several mentors that can give them perspective and help in different kinds of ways. So for example, having a basic science mentor that you may have through your supervisor of your graduate degree work perhaps, but perhaps also a clinical research mentor, and also somebody that you talked about sort of your career advancement with. So some of those things can be covered by the same person, but it's always good to get different perspectives. And I know for me, it's been extremely important having those kinds of different mentors in my life. And when you mentioned self-promotion, for example, was something that I struggled a lot with when I was especially coming to Canada, which was a different culture. I'm originally from Norway, and we have a little bit of a different culture when it comes to, I think, putting your hand up and promoting your own work, and that was something that was very difficult for me. And I think having mentors that pushed me to do it was extremely important for my career development as well. So I think we all have areas that are kind of holding us back, and it can be that, or it could be something else. And having someone that sometimes pushes us, and it can be a little tough love, but that's okay. I think can be really, really useful. So really encourage people to take advantage. And I think one of the things that I regret actually in my career was that when I first got my job as a PI, there was this sort of amazing female scientist that I really admired at my institute, and that I wanted to sort of be my mentor, and I was too shy, and I didn't approach her about it. Looking back, I sort of think that would have been a really good thing to do.
Yeah, no, 100%. I mean, sometimes it's a little daunting at first, but you at the end of the day just can't be afraid of rejection, and I think just going for it and seeing what happens is so important in the field nowadays.
For sure. And if you approach somebody about advice and mentorship, they would be very flattered, right? So I think in general it's not something to be so afraid of, but yeah, it can be very sort of personal and maybe a difficult conversation to initiate if you're more of a shy person. So
right.
But I would encourage people to take that leap of faith.
Yeah. And on that note, kind of what is one piece of advice that you would like to share with students involved in fundamental research who might be discouraged about the prospects of translating their project, or even just in general, who are currently undergoing, studies in basic fundamental research? What piece of advice would you give to them?
Yeah, I would give the advice to, try to, be curiosity driven. If you're curious about a process that, we don't understand how it's working, I think that's when you'll be most successful, if you have sort of a true curiosity and a true passion. So find a question that you're really interested in solving the puzzle, right? Trying to unravel how it all works together. And remember that, there's so many examples of fundamental research that nobody knew how would be applied, but it turned out to be critical. And so I think in either case, whatever you discover will be useful for something, right? It's just that we can't define that ahead of time. If you think of, Watson and Crick and Franklin, right? They were just curious to find out how genetic material was transmitted, and
look how important that ended up being. Yeah,
couldn't possibly know all the applications of really understanding the DNA structure and how that works, right? So I would really say to not be discouraged if you're really investigating something that you're curious about and interested in and passionate about. I think this will be useful somewhere down the line.
Yeah, you never know. You never know the impact that your research might have one day.
It's impossible to know, but that's also a little bit what's exciting about it,
Right.
That, you don't know what that key piece is that you might discover.
Yeah, no. Well, that's some great advice. So thank you for that. And well, that is, I think, takes us to the end of the episode. So thank you again for joining us today, Marianne, and we will see you all in our next episode.
Pleasure to be with you. Thanks for having me.